Okay, good afternoon and welcome to the 2022 Charter Lecture. I'm Jack Hu, Senior Vice President for Academic Affairs and Provost at the University of Georgia. On behalf of the university, I would like to thank you all for joining us for today's 2022 Charter Lecture. The Charter Lecture was established in 1988 to honor the high ideals expressed in the 1785 Charter that made the UGA the birthplace of public higher education in America. Since 1988, we have brought leading voices from around the world to our campus through this lecture series. Today, we hear from two of the UGA's most outstanding faculty members, the 2022 Regents Professors, Professors John Gray and Claudio Saunt. The Regents Professorship is one of the university's highest honors. Those distinguished faculty members will be introduced more fully to you in a moment. In delivering this spring's Charter Lecture, our Regents Professors exemplify the breadth and quality of research and scholarship at the University of Georgia. I invite everyone watching this afternoon to direct questions to the panelists through the Q&A feature. We will curate those questions and present them to our speakers during the Q&A. I'm very pleased that President Moorhead is joining us this afternoon. President Moorhead is a 1980 graduate of the UGA School of Law. He's also the Max Professor of Legal Studies in the Prairie College of Business. Thank you for being with us, President Moorhead.
Thank you, Jack, and good afternoon, everyone. It's my honor to welcome everyone who is attending today's Charter Lecture and join in recognizing UGA's 2022 Regents Professors. I also would like to thank the Provost and his office for sponsoring the Charter Lecture Series. As many of you know, Regents Professorship are bestowed by the University System Board of Regents on truly distinguished faculty members at the University of Georgia. Their scholarship or creative activity is recognized both nationally and internationally as innovative and pace-setting. The two individuals that we have with us today are doing research that is among the best in their fields, and we are certainly very proud of their many accomplishments. They set a bar for excellence that is helping UGA as we continue to seek to be among the best research universities in the entire nation. Interdisciplinary research is one of the hallmarks of UGA's research enterprise, and I'm delighted that this year's Charter Lecture features research at the intersection of history and infectious disease. Dr. Saunt and Dr. Drake will discuss their individual research programs and their collaborative scholarship on smallpox in early America. Over the years, these outstanding professors have helped UGA build a strong culture of research mentoring, and you will hear more about that from the students that will introduce them. Undergraduate student Thomas Russell will introduce Dr. Saunt, and doctoral student Anna Willoughby will introduce Dr. Drake. So thank you so much for being with us today, and I'll turn things over to Thomas and Anna.
Thank you so much, President Moorhead, for your remarks. And it is my pleasure to introduce Dr. Claudio Saunt. Dr. Saunt holds the Richard B. Russell Chair in American History, as well as the title of Distinguished Research Professor. He is the author of a wide breadth of books and articles on early American history, and has distinguished himself as a foremost authority on the history of Native Americans in the American Southeast. His most recent book, Unworthy Republic, sheds light on the tragic story of Indian removal of 1830, and was recognized nationally as the winner of the Bancroft Prize in American History, the Robert F. Kennedy Book Award, and the Rittenauer Book Prize. His other works, including A New Order of Things, Black, White, and Indian, and West of the Revolution, focus on everything from Greek history, the complexity of early American racial classifications, the American West during the Revolutionary War, and more. No matter the topic, Dr. Saunt's keen eye for research and attention to detail has remained a central tenet of his scholarship and has earned him praise and recognition from both his academic contemporaries and the casual history reader alike. Of course, I know Dr. Saunt is my thesis supervisor. I can't begin to count the number of times he's pointed me in the right direction for a research question or gave me well-meaning critiques on an aspect of my thesis. He continuously makes time for his students, both his graduate students and his undergrads, and he commands the respect and admiration of my peers. In my view, Dr. Saunt embodies what it means to be a teacher and an educator. Again, it is my pleasure to introduce Dr. Claudio Saunt.
Thank you, Thomas, for that very generous introduction. And it's been a real pleasure working with you this semester. I'm looking forward to seeing your honors thesis. So I'm going to talk about a little bit about my latest book today, Unworthy Republic. It's about Indian removal, when the United States under President Andrew Jackson deported 80,000 Native Americans from their homelands east of the Mississippi River to the area that became Oklahoma. I make three arguments in the book. First, there was nothing inevitable about this moment, despite what we may think. And depending on your political perspective, you might believe that Native peoples were too primitive or backwards to survive in the shadow of the expanding United States. Or conversely, you might think that white people were too avaricious to let them remain on their homelands. But in fact, the vote in Congress came down to 5 votes out of 199 cast. And this was a hotly contested vote. It came down to the wire. The second argument I make is that this was a turning point. We can think of it as the Great Divide, when the United States demarcated a border between white and Black Americans on the one side and Native Americans on the other. And third, I argue that this moment ought to be understood as one of the first state-sponsored mass deportations of the modern era, creating a model for other modernizing states around the world. But today I'm not going to elaborate on that story. Instead, I'm going to briefly describe what kinds of evidence I use and what historians do. Simply put, we historians write narratives about the past, but we do so relying on a set of conventions. And the most peculiar and salient of those conventions is the footnote that refers to archival materials. This is a kind of field marking that identifies historians in the wild. So using the examples from my recent book, I'm going to briefly describe what those footnotes do and what kinds of evidence they refer to. Historians traditionally rely on written records, though that has changed over the past generation as we've incorporated oral traditions, linguistic evidence, material culture, that is physical objects, and also archaeological evidence. So in this instance, I was especially interested in the role of the federal government and its bureaucracy in administering the deportation of 80,000 people in the 1830s. So here you can see a letter from the Commissioner of Indian Affairs to Joel Poinsett. This was the Secretary of War, who was also a wealthy slave owner from South Carolina. This document was written in 1839, and it's in the National Archives. This is an example of high-level correspondence. It's useful for understanding state policy, and it revealed to me how federal officers based in Washington, D.C., had very little idea what they were doing. They did not understand the people they were deporting. They did not know where exactly they were moving them. And they struggled to get a handle on the logistics of the operation, to get food and other supplies to the right place at the right time. So these kinds of letters were extraordinarily valuable, but they're also very far removed from what was happening on the ground. So for that view, I had to look elsewhere. So here's a letter from a land speculator in Pontotoc, Mississippi, which was then the Choctaw Nation, written to a banker in New York City. And here you can see in this excerpt, he says that this company of land speculators based on Wall Street has fixed a minimum of a 200% profit on Indian lands. So this letter and others like it revealed a part of the story that was largely unknown. That is, we think of Indian removal largely as a Southern story, but much of the financing came from Wall Street. So this example comes from the Wisconsin Historical Society in Madison. Other sources are scattered around the country. Here's a diary. This is from an officer named Nathaniel Weish Hunter, who was fighting in the Second Seminole War, so in the second half of the 1830s. And the diary is located in Mills College in Oakland, California. So here's a diary of an officer named Nathaniel Weish Hunter. As I mentioned, he fought in the Seminole War. And here he writes about arriving at Fort Cummings in Florida. Fort Cummings was named for a lieutenant colonel named Alexander Cummings. And you can see in this excerpt here, he says that the fort is a fit tribute to such a man. Happy would it be for his men that this would be the only memorial to perpetuate a name linked with no virtue and 1,000 crimes. Hunter, by the way, was married to an Athens woman, and he's actually buried right here in Oconee Hill Cemetery. So one of the challenges is to capture Native voices, and sometimes we can do that by scouring the archives and finding letters such as this one. This is a petition from a group of Potawatomis, Ottawas, and Chippewas written to Andrew Jackson. And here they write, "Father, we have been disappointed and dissatisfied. We are told by their fellow countrymen, other Potawatomis and Ottawas, we've been told that our new country is mostly prairie, that there is scarce timber enough to build our wigwams, and that some of our land is too poor for snakes to live upon." Other Indigenous leaders accused Jackson of hypocrisy and suggested that he was a despot. So in the case of the Cherokees, we can use the Cherokee Phoenix. This is an extraordinary resource. It's the first newspaper that was printed by an Indigenous nation anywhere in the Americas. Here is a memorial that was printed in the Cherokee Phoenix in 1829. And what's fascinating about this is that there's an English translation on the right side of the page, and then it exists in Cherokee, in the Cherokee Syllabary on the left-hand side. And you can see it, just a close-up here of these beautiful Cherokee characters. This syllabary was invented in the early 1820s. So I had the Cherokee part retranslated because I was curious if the language, if the two versions of this petition to Congress would actually say the same thing. And interestingly enough, there are some telling differences. So in English, in this appeal to Congress, the memorial gently states that the Cherokees had a number of grievances with the federal government. But in the Cherokee language, the Cherokee version, the same sentence flatly asserts that the United States had treated them badly and cheated them. So in other words, there are two versions of this memorial, each written for a different audience. At times, the document itself, rather than its content, becomes a source of information. So this is a receipt or an invoice in and of itself. It's not particularly interesting. It says $10 paid to William Spickneagle for making a coffin for a Cherokee leader named Okefuscaholo in October of 1836 near Memphis. But there are tens of thousands of invoices and receipts like this one held in the Treasury Department. And they reveal how this massive experiment with the lives of other people became an accounting exercise for many clerks working in the federal government. Stacked one atop the other, the accounting records would surpass the height of the Statue of Liberty. I mentioned at the outset that not all of the sources are textual. And since many Native peoples were not literate, archaeology can help us fill out a picture of their daily lives. Here you can see some deer and turkey bones that were recovered from a lower creek town, so in present-day Alabama. And they tell us about subsistence practices. Just as interesting, archaeobotanists have recovered plant remains that reveal which plants Native Americans relied on for food and medicine, which essential medicinal sources they left behind when they were forced west. And finally, new technologies help us use archival sources in innovative ways. So geographic information systems allows us to store information spatially. It allows us to see spatial patterns that would not otherwise be evident. So here is a list of Chickasaw land purchases by a group of speculators, once again based in New York City on Wall Street. And this is a list that goes on for pages and pages. On the left-hand column, there it lists the township and the range and the section number. On the right-hand column, it lists the number of acres in each one of these land purchases. So only 20 years ago, it would have been difficult, if not impossible, to make much of this list of these hundreds and hundreds of tracts of land. But now with GIS, we can plot all of these on a map. And that's what I did here. You can see on the left-hand side, all of these black squares represent their land purchases. And then we can overlay that with other kinds of data. So here you can see this highlighted polygon, which represents the Chickasaw Nation in the 1830s. And then I overlaid that with data collected from a NASA satellite image showing the Black Prairie lands. This is this arc of very fertile soil that was formed in the Cretaceous era, 70 or 80 million years ago. It's extremely fertile, rich in calcium, perfect for growing cotton, and it was some of the most valuable agricultural land in the entire world at the time. And so we can see that these land purchases by these speculators based in New York City were really concentrated on these soils. The Black Prairie you see here, and then this other area which is highlighted, which represents also lands which are suitable for cotton cultivation. So you can see the kinds of evidence that I accumulated for this book. One of the challenges in the discipline has been to move past textual evidence. And while we've done a good job incorporating material culture, archaeology, and anthropology, we've not done so well using some of the tools that ecologists and population geneticists have at their disposal. So John Drake and I will talk about that in the third part of this talk. But in the meantime, I want to turn it over to Anna Willoughby to introduce John to you.
Thank you, Dr. Saunt. It's a pleasure to introduce Dr. John Drake. Dr. John Drake is a professor in the Odom School of Ecology and director of UGA's Center for the Ecology of Infectious Diseases. He's conducted research in the interdisciplinary field of population biology, crossing boundaries between ecology, evolutionary biology, and epidemiology. With 182 peer-reviewed publications, Dr. Drake is a globally recognized scientific authority on the population biology of infectious diseases and has done groundbreaking work to model early warning systems for both population extinctions and emerging epidemics. Dr. Drake can make small things powerful. His experimental studies of zooplankton illuminate the key processes that regulate populations like temperature, environmental noise, and extreme weather. He also leverages tools from physics and mathematical modeling to understand the spatial dynamics of biological invasions, from fungal spread in North American bats to Ebola virus in West Africa, or aquatic organisms spread through worldwide shipping networks. To recognize his outstanding contributions, Dr. Drake has received a number of honors. Within UGA, he was awarded a Creative Research Medal in 2014, followed by the title of Distinguished Research Professor in 2017. He is a fellow of the American Association for the Advancement of Science and the Ecological Society of America. And recently, Dr. Drake has expanded his scientific audience by contributing to Ford's, writing accessible pieces to an audience of more than 300,000 online readers. Not only is Dr. Drake tremendously successful in his own right, he is also dedicated to the success of those around him, including his own students and postdocs and his colleagues. Through NSF and NIH-funded training programs, he's advocated for the next generation of ecologists. His NSF REU program in particular has hosted more than 100 undergraduate students to date. Ecologists are challenged to balance generalism, realism, and precision in the world's complexities. As one of Dr. Drake's doctoral students, I've seen how he tackles this challenge with robust, innovative, and purposeful science. He develops models that can advance both ecological theory and provide applied solutions. I experienced this the most while collaborating with him in the center's coronavirus working group, where we created predictive models for SARS-CoV-2 transmission with different interventions and provided situation awareness through new data visualizations. My favorite part was seeing Dr. Drake rally his colleagues and students across campus to work together towards a common goal. In sum, Dr. Drake is an accomplished and creative scientist, a strong collaborator, and a supportive mentor. And so with that, I will turn the program over to him.
Thank you, Anna, for that kind introduction. I've also enjoyed our time working together. So, Claudio is a historian, and I'm a scientist. Preparing for today's talk really caused me to reflect a lot on the role of method in scholarship. It's true that there are differences between history and science, and that these differences are deep. In preparing for the talk, I tried to come up with a short list. I don't think I did a very good job, but here are some possibilities. So history is exquisitely concerned with the particular, whereas often science strives to be as general as possible. History is personal, it's about us, while science is about that, the other wonderful things in the universe, like molecules and viruses, cells and organisms, volcanoes and galaxies, things that are not us as we understand ourselves. Historians make judgments, scientists make measurements. Everything the historian studies is one-off, a string of happenings in some place at some time, whereas repeatability is a much praised virtue in science. Perhaps the role of theory, whatever that may be, is something that distinguishes history from science. But in fact, I do not think this is a very defensible list. The Human Genome Project was all about us. In fact, all of biomedicine is about us. And there are many non-experimental areas of scientific inquiry that deal with one-off happenings, from planetary formation to biological evolution. So maybe it is more interesting to focus on the similarities. For instance, both history and science are deeply concerned with empirical evidence. I think that's a lot of what Claudio talked about just a few moments ago. To be accepted in science and in history, explanations are accountable to data, facts, and observations. Both history and science are examples of what the philosopher of science, Bas van Fraassen, calls objectifying inquiry. Historians are trying to figure out what happened. I study the ecology of infectious diseases and the dynamics of epidemics. When we study epidemics, we're trying to figure out what happened. For instance, here are some data. These are data related to the ongoing COVID pandemic that I downloaded from the Georgia Department of Public Health website. These data show the reported number of positive cases of SARS-CoV-2 infection. We, and many others, both historians and scientists, are trying to figure out what happened. We call a figure like this the epidemic curve. The data expressed in this graph are some of the facts that have to be accounted for by any satisfactory explanation of SARS-CoV-2 transmission. One interesting difference between historians and scientists is how we express our ideas about what happened. Our narrative styles are different. For instance, scientists use mathematics to describe the world, not exclusively, of course, but to a much greater extent than historians. So here's some mathematics. A system of 3 equations, which together constitute a model. The way the model works is this. First, we classify everyone in the population according to 3 mutually exclusive categories. Susceptible people, represented by the variable S, are those without the infection and vulnerable. Infected people, denoted by I, are those with active infections. And people who are recovered and immune are designated by R. S, I, and R are known as the state variables because they describe the state of the population at any point in time. During an epidemic, of course, the state variables are changing. Susceptible people are infected, infected people eventually recover. So we think of flows between the compartments. In this system of equations, the flows are given by the processes of transmission and recovery. Transmission and recovery occur at rates that depend both on the number of individuals in relevant classes. For instance, if there are a lot of infected people and a lot of susceptible people mixing together, then the transmission rate can be expected to be high, as well as other quantities represented here by the Greek letters beta and gamma, which are known as parameters. In this model, the parameters are fixed constants that describe the speed of transmission and the speed of recovery. From the diagram of flows, we can write down differential equations that describe the rates of change of S, I, and R with respect to time. Because S, I, and R appear in the equations for each other's rate of change, we have something that can be thought of as a system of interacting parts. When equations are presented in this abstract way, unattached to anything particular in the world, we call it theory. A theoretical biologist then asks what kinds of phenomena are consistent with this picture. To answer that question, we solve the equations. And here are some solutions. What you see is that, in this model anyway, the population of Georgia starts with everyone susceptible at the beginning of the epidemic. This is the blue line. The I variable is indicated by the red line. And when we see a wave in the red line, that is an epidemic. And the green line shows the total number of immune people in the population. To present these solutions to you, I must particularize a bit more, however. I have to specify an initial condition and values for the parameters. But having done that, I now have a device that can be compared to the epidemic curve. A good explanation is one where there is a good match between the equations and the phenomenon. As a so-called modeler, my job is primarily to come up with the equations and figure out the details to achieve a match. When the match is poor, we conclude that some part of the explanation is missing. We quantify this fit using statistics. Thus, mathematical statistics helps us to understand what we mean by a good match. Computational statistics tells us how to find the explanation that best matches. So let me continue with the example from the COVID pandemic in Georgia. Here again, we have the epidemic curve for cases in Georgia. For comparison, I show the solution to the I equation. An interesting feature of the data is the multiple waves of COVID-19. One reason this is interesting is that the equations I presented earlier don't admit multi-wave solutions. So that means something is missing from the explanation. These data have other more subtle characteristics that don't match those equations too. For instance, the speeds at which the waves advance and recede. When we look at other bits of data, like the number of deaths attributed to COVID-19, we find even more ways in which the explanation offered by these equations is inadequate. The SIR equations are not a good model of SARS-CoV-2 transmission. Of course, that's not surprising. We all know that SARS-CoV-2 transmission is a more complicated matter than can be expressed by 3 equations. Although it is remarkable how so often such simple equations do explain so much. Perhaps you noticed that when I described these equations, I didn't mention anything about physical distancing, wearing masks, vaccines, and so forth. Epidemics that follow these equations end due to what we call susceptible depletion. The epidemic burns through the susceptible population. If prior infection results in immunity, then the stock of available people that fuels the epidemic is reduced over time. Susceptible depletion has occurred during COVID, but not to a sufficient level to result in containment. The earlier equations also don't account for asymptomatic transmission, a lag between infection and symptom onset, a changing standard of care as we've come to better understand the disease that SARS-CoV-2 causes, and a host of other factors. Thus, during the early days of the pandemic, a group of us in the Center for the Ecology of Infectious Diseases developed a new model and strived to continually update this model as more information came out. By the end of the first year, the model looked like this. Instead of 3 equations, we had 17. The model is also what we call semi-parametric. This means that there are factors that we know are important, like physical distancing and mask wearing, that we don't have enough information to include directly in the model. But we can create a variable that's a placeholder for these unmeasured things, and using some new statistical technology, estimate what their magnitudes must be. Scientists often measure unseen forces based on their effects on other things. Dark matter in the universe has never been observed, but is believed to exist because observations we make only make sense if there is such a thing, along with its associated gravitational forces. This is a picture of what we observed during 2020 and 2021. On the top, you see the case reports over time. In the middle, in blue, you see more data that has to do with how people moved around. The nosedive before April 24th, 2020, when the country started to reopen, had a very real effect. I believe it prevented us from having a very rapid summer epidemic that would have completely overwhelmed our hospitals. The green line is the dark matter, the unobservable part that measures the effectiveness of other control measures, like mask wearing. Both of these factors, along with intrinsic properties of the epidemic, like the changing immune profile of the population, give rise to the effective reproduction number plotted in the bottom panel. The effective reproduction number provides an overall summary of the propensity for the epidemic to grow or decline. It's one of the key quantities that we studied as the epidemic was unfolding. The last thing that I'd like to point out is that studies like this are not simply academic exercises. Throughout the COVID pandemic, public policy decisions had to be made. I believe that data-driven epidemic models like these can be very useful in making those decisions. In this case, we operationalized our model to investigate alternative scenarios related to our mobility patterns. How would it affect the epidemic if we resume shelter in place? What if we completely return to pre-COVID levels of interpersonal contact? These are the kinds of questions that epidemic models allow us to think through. The overarching goal of my research is to help us to do that better in the future. And so now I'd like Claudio to mention our work together, and then I'll follow up and tell you about some of the analysis that we've been doing.
Thank you, John. So John and I met several years ago at a Wilson Center event, and we recognized immediately that we had overlapping interests. I've long been interested in the demography of early America and had launched a project to map the African, European, and indigenous populations in the colonial era. And John has expertise in population dynamics and species distributions. So this led to a collaboration to model the distribution of the native population in North America between 1500 and 1800. That's an ongoing project. More recently, we began working together to model the impact of smallpox on native communities during the same period. This is a pressing question in my discipline. For half a century, viruses have been central to our understanding of the colonization of the Americas. Beginning in the 16th century, conquistadors and other colonists carried novel viruses across the Atlantic and unintentionally introduced them to indigenous communities. Smallpox, the deadliest of them, killed up to 90% of the native population. So the argument was simple, perhaps too much so, but let me describe it to you briefly. When novel viruses are introduced to a population that has never before been exposed, the effects can be devastating. No one has any acquired immunity. Much as COVID-19 raced around the world, so too smallpox swept through the Americas. In Europe, Asia, and Africa, by contrast, the virus appeared frequently, and over time, a large percentage of the population had acquired immunity. Once infected, an individual either dies or recovers and is immune for life. So as a result, if smallpox appeared every 5 years, say in an English village, only those born since the last outbreak were susceptible, with only a few exceptions. But in the Americas, everyone was potential food for the virus. Communities that were struck by the microbe sometimes collapsed. Not enough people were healthy to gather firewood, to collect water, clear fields, or harvest crops. Elders died off. They were the repositories of centuries of accumulated knowledge about where and when to fish, where to hunt and plant. They were also the guardians of cultural traditions. But recently, historians have questioned this narrative, raising 3 objections. First, far from having a uniform impact on Native peoples, novel diseases affected each community differently, depending on numerous factors, including the date of first exposure, environmental conditions such as humidity and temperature, the season of the year, which determined the volume of local food surpluses, and the kinds of labor that people had to perform to sustain their families, the response of community members to the illness, their daily pathways, and the time between outbreaks. The second objection is that the assertion that this disease, that smallpox wiped out 90% of the indigenous population, is inaccurate. No single epidemic of any known microbe has such high overall fatality rates. And between exposures to this virus, birth and death rates will change the underlying denominator, the size of the population. So while it's entirely possible that the nadir of the indigenous population in North America, that is approximately a little over 500,000 people in 1900, it's entirely possible that that number is only a 10th of what it was 4 centuries earlier. That decline reflects the cumulative effect of birth and death rates over 20 generations, with epidemic disease representing only one of many factors. And the third objection to this narrative is that by dispossessing Native peoples, by destroying their subsistence base, scattering their communities, and waging war against them, colonists created the very conditions that led to such high population losses. Some scholars have gone further, suggesting that the focus on microbes has provided a convenient way for European Americans to escape blame for the genocide that occurred in the New World. In other words, we didn't do it, the virus did. And so it is now no longer clear what role novel viruses such as smallpox played in the colonization of the Americas. And so I approached John last year, not coincidentally in the midst of the COVID-19 pandemic, to see if we could collaborate on a project to move the scholarly debate forward. The historical sources in this era are limited, and we will not discover others. There were no epidemiologists keeping mortality tables in the Cherokee Nation, and demographic data are scattershot and not always reliable. So to move forward, we need to draw on some of the innovations in disease modeling that John and others have made over the last generation. I asked John some basic questions. First of all, how would the transmissibility of the virus affect the overall mortality rate in a village of, say, 1,000 people? How would the population recover over time, given specific birth and death rates? What would happen if the virus were reintroduced every 10 years or every 20 years or every 30 years? And finally, how would village size affect the overall mortality rate and population recovery? And so I will turn it over to John to provide some of the preliminary answers to these questions.
And so I thought, these are the kind of abstract questions that we can use epidemiological theory to answer. How fascinating that their answers are of such interest to a historian. As we discussed more, I came to realize that the core question being asked was one that theoretical epidemiologists had never fully asked before. The question was this: imagine we have a village of 1,000 people. For epidemiological purposes, the village is isolated. It's its own demographic entity. And should there be an epidemic, the epidemic will happen quickly within the context of that village. A scientist would say that the dynamics within the village are fast, while the dynamical connections between villages are slow. This separation of timescales allows us to focus just on one village, so long as we represent in some way that it is weakly connected to other villages, allowing the possibility of the introduction of the pathogen on some relatively rare occasions. Then the question is, how are the long-term prospects for population growth and decline over multiple generations affected by the combination of repeated introductions of a lethal disease and the intrinsic capacity of the population to recover from these demographic perturbations? Theoretical epidemiologists have long had an interest in epidemics in systems comprised of coupled populations. This is called metapopulation theory. But most studies of epidemics in metapopulations are concerned with the theoretical properties of the entire collection of villages. Here, we wanted to understand a single village in that context. This had not really been done before, at least not in a way that would answer Claudio's questions. Our first step was to create then a new model. The model is similar to the other two models I've already shown today. It consists of 3 differential equations, but there are some important modifications. 2 of these modifications are familiar and have been studied before. These are the introduction of demography, births and deaths, in a way that is allowed to be out of balance. The second is the introduction of disease-induced mortality, because smallpox is so often lethal. To my knowledge, the third modification is new. The third modification is to allow there to be spontaneous infection at some low rate. That is, a disease-free population has the disease introduced by someone not from that village. That part had actually been done before. The new part is we wanted that rate to stay the same regardless of the village size, which will be fluctuating due to demographic changes and disease. The idea is that if your position in the larger network of villages is such that potentially infectious encounters occurs X times per year in the course of trade and travel, that should not change just because the population grows or perhaps is reduced by half or so in an epidemic. Of course, this is an idealization, but it is an important new idea which we have incorporated into the force of infection. So here's our model. There's another thing I need to say at this point. Some of you in the audience will know that equations like these typically give rise to unique and smooth solutions. But populations, especially small populations, don't actually change in smooth ways. And we don't think that the future of any population is essentially deterministic. So for our hypothetical village of 1,000 people, we actually interpret these equations as the average over a number of possible scenarios that are themselves the realizations of a probabilistic process. For today's purpose, there's no reason to go into the details of how we do this, but that background is needed to understand the results that I'm presenting. Armed with this model, we can now investigate some scenarios. In the first scenario, we assume that the village of 1,000 people is in demographic equilibrium prior to the introduction of smallpox. We also assume that smallpox is introduced on average once a year and that 55% of the infected population succumbs. This plot shows how the population size of the village changes over time. In each simulation, introduction of the pathogen results in a large initial outbreak, reducing the population substantially. Because most of the population has either been killed by the virus or made immune, subsequent introductions only cause very small outbreaks. We even hardly see those outbreaks in these data. Still, because the population was in demographic balance in the absence of smallpox, these small outbreaks still lead to a continuous decline in the population. So at the end of 200 years, the total population is only about a quarter of the initial size of 1,000, shown by the histogram on the right-hand side. But, of course, not all populations are in demographic balance. In populations with positive growth rate, there is the possibility of rebounding from a demographic perturbation caused by an epidemic. Well, we studied this in a second scenario. In this case, because the population is resilient, it quickly builds up a pool of susceptible people, which then is subject to large outbreaks every 10 years or so. In this example, the intrinsic ability of the population to increase in size means that the net change in the population over 200 years is growth, increasing from 1,000 at the beginning to around 3,000 by the end. In this figure, I'm showing just one realization. The black line on the y-axis on the right shows the total population size. The red line shown on the left is the number of infected people. And we see a pattern of recurrent outbreaks. What these two scenarios point to is that, in fact, there's a balance between the intrinsic potential for reproduction and the frequency of smallpox introduction. So in one final analysis, we simulated 10 populations for lots of combinations of reproduction rates and smallpox introduction rates. And to summarize the impact of smallpox on population size, we calculated the ratio of the final population size to the initial population size as an index of the severity of the epidemics on the population. A value of 1 for this ratio is a population that doesn't change in size, and values less than 1 are populations that declined, while values greater than 1 are populations that increased. This figure shows the results of that analysis. Let me orient you to the figure. The demography is on the x-axis, expressed in terms of the number of births per capita per year. And the rate of smallpox introduction is on the y-axis. In principle, if you had information about the demography of any particular village or people group, you could locate them on this figure and ask whether or not the theory matches the observation. Societies to the left of the contour line at 1, that's the contour line that runs down through the middle of this plot, are those that would have declined in size due to smallpox. Those to the right are those that would have increased, and those right on that contour line are the ones that would have stayed the same. At this point, this is basically the state of our research. From experiments with these models, we've concluded that figuring out the basic underlying demography of early American populations is really the key to understanding how much smallpox introduction shaped their decline. What we're doing now is taking a deep dive into the existing literature on early American demography with the hope of mapping real populations to this space. It's our hope that when we've done this, we'll have a much clearer understanding of the actual role of infectious diseases during the European expansion in North America.
Okay, thank you, John and Claudio, for the very interesting presentations, including your collaborative project. So I will now invite our audience to pose questions for our panelists through the Q&A feature. So you can type in the questions, and then I will read those, and then either John or Claudio or them together will try to answer your questions. So while we are waiting for questions to show up in the Q&A, maybe I can start. So the two of you, Claudio and John, presented a very interesting collaboration, and your collaboration really are across disciplinary boundaries, history and infectious disease and ecology of infectious disease. So what advice do you have for other researchers who are exploring opportunities for collaboration across discipline boundaries? So advices that you may have for faculty and scientists or historian or other who are exploring potential collaborations.
I can offer some very practical advice, which is that these sorts of collaborations are dependent on sending emails and asking questions. And in universities, we're so often in our own little departmental silos. It's infrequent. I think that we do reach out, and as I mentioned earlier, I met John quite by accident, and we just struck up a conversation and started talking. But I do think that kind of just curiosity and sending an email to someone in a different department who may be able to help out or shed some light on questions that you might have is a necessary first step, and then it can evolve from there.
I fully agree with that. And curiosity really is, I think, an intellectual virtue that oftentimes in the busyness of our disciplinary scholarship, we fail to cultivate. And so taking that curiosity and really engaging with it. I've enjoyed a great deal my conversations over lunch with Claudio about the questions and methodology that his discipline is asking, the questions I didn't even know how to ask. And so that curiosity goes beyond Claudio sending a handful of questions that he thinks theoretical epidemiology might help to answer. But I had to return that curiosity by asking, well, what's been done on these questions before? How are these questions thought about by your discipline now? Teach me about what you know, what your methodology is, what the history of the conversation to date is, so that I can understand that in a more fully formed way.
Very good, thank you. Maybe I can have a follow-up on this particular question. So your random encounter through some events in the Wilson Center help promote the collaboration. So have you encountered some challenges in your collaboration? For example, John mentioned the different methods to research and scholarship. So were you able to have some common methods or common languages for your collaboration?
I think it helped that, or I assume it helped that John, I believe, majored in philosophy as an undergraduate, and he was open to the kinds of questions and used to speaking in the kind of language that historians do. So that certainly made it easier. Now, I will just add an asterisk that we are in the midst of working on this article, and we've yet to actually write something. And I think that, well, we do have some language on a piece of paper, but I think when we're actually kind of working through how we structure and shape the argument and present it and choose the kinds of language that we want to use, that I think I expect we will run into some challenges there, but I'm certain that we can overcome them.
I agree with that. Negotiating language takes a lot of work. And I think it was many conversations before we had what we understood to be an answerable question for each of us. But that was part of the discovery process. That was part of the curiosity. Also, it's the case that some of the sources that Claudio pointed me to used, to some extent, demographic methods that actually were developed in the context of ecological studies of wildlife and human demography decades ago, and had already been applied by demographers and historians to understanding the demography of early American populations. So some of that work was already done for us, I think.
Okay, thank you. Let's go to the chat box. Is smallpox the only disease imported from Europe that is thought to have impacted the native populations? If it's not the only one, have you considered models that combine impacts of multiple disease?
It's certainly not the only disease. Measles, influenza, yellow fever, malaria, and many others. Smallpox, I think, had the largest impact. We did discuss early on incorporating measles, but first things first, and we will see if it will be possible to, or even necessary to incorporate measles to answer some of the questions we're asking. I think we'll just see how that unfolds in the future.
Okay, anything to add, John?
So I agree with that. We certainly have empirical evidence that other diseases had large impacts on both European Americans and Native populations. But my approach usually is to start with the simplest system that we can and ask whether or not the theory that we develop, as I say, matches the observations that we have. We discovered quickly that there were some new contributions that we had to make, models that hadn't been written down before. And working out those details is prerequisite to, for instance, bringing in multiple factors. And one of the things I like about my discipline, ecology, is how holistic it is. I often say that this is really one of the hallmarks of ecology as a science. It aims to be both holistic and reductionistic at the same time. What that means is we spend a lot of time teaching ourselves and our students to try and come up with exactly the right representation for the complexity of the phenomenon that's of interest. We take care not to overly simplify our theory, but at the same time, we don't want to introduce unnecessary distractions, which then makes it very difficult to parse out what the important causal factors are. I think we're trying to adopt a similar kind of philosophy when we approach this question. But as Claudio says, it remains to be seen how well that works.
Okay, thank you. Next questions. Thanks to Dr. Drake and Dr. Saunt. On the next steps in the smallpox project, what kinds of evidence and model is the literature relying on to determine birth rates in early American communities?
Yeah, that's a great question, and I think what it's pointing to is that the demographic data is unreliable, and we're collecting that data right now into a spreadsheet. But I mean, we're relying on the kinds of evidence you might expect. There's archaeological evidence based on burials, and then there is and population growth rates over time, the size of the village settlement, and then there's also documentary evidence beginning in the 17th and 18th centuries. But again, those numbers, these are not scientists or demographers. These are frequently explorers or traders who might mention simply in passing something kind of general about the birth rate of a given population. So, I mean, what we're going to do is compile the kind of range of possible birth rates. I mean, one of the advantages of working with someone like John, who's a modeler, is that you can plug in all kinds of different variables and just see what happens. And so that's what our intention is. We're not going to choose a single birth rate and suggest that somehow that's a reflection of what was actually going on at a specific time and place.
And I'll add that there are techniques that we can use to try and back out some of the information that we're interested in from things like lifespans, family size, the age structure of a population, which sometimes has been recorded by early explorers and early observers. Also, of course, being interested in the use of statistics to match evidence with ideas. One of my goals is for us to treat this in maybe a more rigorous fashion than has been done before. So we'd like to put some uncertainty around what we think the possible growth rates of relevant populations are, and then ask how does that range of possibilities coincide with what our model tells us would be the long-term outcomes for population growth or decline.
Okay, thank you. Next question, I think is direct to Claudio. So Claudio, can you compare the concept of theory as it applies in history with scientific theory?
I think John the philosopher should answer that one. I mean, I would say that historians deal in narratives, scientists do too, except that our narratives are all about contingency. So we're not trying to model the natural world or even human behavior in any way. Contingency is really fundamental to what we do. And of course, there are no controls either. But since we're not building models, with a few exceptions, that's really not a concern of ours.
But I think it's mistaken to think that scientists are not interested in narrative. We just use different language to talk about narrative. Often the language is mathematics. Now, why do we use mathematics? Because it's a way that allows us to make our ideas clear. And that clarity then allows us to do this comparison of evidence and idea. I think that there's actually more promise for using the kind of mathematical models that we use in ecology and infectious disease dynamics to answer questions in other fields of inquiry, including history, political science, and others. There was a movement, I think, in history a couple of decades ago called quantitative history, in which questions about historical events that might be more traditionally answered in a narrative fashion are related to quantities that are measurable. I remember when I was a graduate student, I attempted a term paper along these lines. I was interested in a debate in the sort of early decades of my field about what counted as being in and what was out. And there were questions about who had the authority in the field to decide that, journal editors and so forth. And as a result, the Journal of Animal Ecology was started. And I went back to the early tables of contents of the Journal of Animal Ecology and scored the titles of those papers according to various criteria in an attempt to quantitatively answer the question, did this journal introduce a disruption to the field that was initially claimed or not? Or was it just doing the same thing that that in this area of science had already been done? They were just doing more of it. I think that's a genuine historical question. Maybe it's not of interest to a wide range of people, but it's one that I thought that could be answered using the same kind of statistical analysis and hypothesis testing.
Next question. Thank you for the clear and convincing positives in collaboration, Dr. Drake and Dr. Saunt. I'm curious whether the finer grain and more flexible approach of your partnership shows combining qualitative and quantitative approaches might answer not only historical questions, but also be helpful in shaping public behaviors in the next pandemic.
So I think that there's
not only help explaining what happened, but can you also be of guidance in public behaviors in the future if we are faced with a pandemic?
I think this is one of the things that COVID-19 has really exposed. Of course, the lack of knowledge about risk perception, cognition, human behavior in response to crisis events. That's scientific content that many people are working on now. But there's also the methodological question of how do you actually integrate information, ideas, conceptual framings that come from the social sciences with methodology, data, and theory that come from the natural sciences. It's something that in the Center for the Ecology of Infectious Diseases, we are actively working on, and I expect we'll be pursuing, pretty intensively in the future years.
Yeah, I mean, these are questions obviously that public health officials have been wrestling with for generations. I mean, I can just say personally that teaching about disease in early America and Native American history over the last couple of years has certainly seemed more relevant and urgent, and that I think that it does reach the students. They certainly connect the dots between history in the 18th century in North America and what's going on in their own lives today.
Okay,
You know, that
thank you.
historical perspective also might be really important for us to emphasize both in our classes, but also in the larger cultural context. And what I mean is that most of us alive today were born in an era where we had antibiotics and vaccines. And so the importance of infectious disease in human society has been mitigated to a large extent by medical advances. But when we have emerging infectious diseases, it takes time for those medical advances to be developed. And in some ways, we're back to the pre-vaccine era. We're back to the pre-antibiotic era. And our interventions are behavioral. And so I think we should not lose sight of that fact, especially in light of the many changes that are going on around the world that imply that emerging pathogens are likely to be more frequent in the future than they have in the last few decades, where they've certainly been frequent enough as it is.
Very good. Next question. Can the science of DNA be used in the kind of research that you are doing collaboratively? For example, are there DNA markers for survival of smallpox?
I haven't seen any such studies. I'd be surprised if that such studies, you know, I don't think that's likely to be the case, that we'll find such markers in DNA, but who can say? But what has been useful, and which is a growing field, is to use DNA to estimate early population sizes. There was just a fascinating article about the early population in the Caribbean, and this has been a hotly debated question. What was the population in the 16th century at the time of contact? We have very few written sources, but we now have some interesting DNA evidence suggesting that the number was far lower than we had thought. A lot of that work is being done by a population geneticist at Harvard's Broad Institute named David Reich. And I've actually begun collaborating with him on a project to better understand the early population in the US South. So I think there is some exciting work to be done in the future there.
I agree with Claudio about that. I think that the use of molecular biology in history, for instance, is likely to increase. I think that many of those uses will be indirect. They won't directly answer our questions. But for instance, by looking at genetic patterns in contemporary populations, we can sometimes infer things about growth rates or population size. Also, technological advances in the last decade mean that there are paleomolecular studies, studies that allow us to extract ancient DNA and learn about the genetics of populations that existed hundreds or thousands of years ago. And I think both of those uses of molecular biology in historical inquiry, archaeology, and so forth are likely to increase.
Thank you. I have a question for John here. John, since July 2020, you have used your research expertise on infectious disease to write numerous public science articles, in particular in the Forbes magazine. So has this writing for the public impacted your research or scholarship? Has it changed your views about what you're doing in science?
So this has been an experiment for me. I haven't been that kind of writer before. And I've really embraced the challenge, although I don't think I'm very good at it yet. It has changed not the kinds of science we do or the way we go about our science, but one of the things that it has made crystal clear for me is the difficulty of expressing the questions that we have. And so the questions that people have asked me that I've tried to write about are often very straightforward. And I think that finding the answers to them is not at all straightforward. And so learning something about asking questions, I think, is something that I've learned from that exercise, and hope will continue to practice in the future.
Okay, thank you so much. I think we have come to the end of our time and also to the end of our questions and discussions. I want to take this opportunity to thank Claudio Saunt and John Gray, our two newest Regents professors, for their remarks and for their dedication to scholarship and teaching and service. Of course, I would like to thank President Moorhead for being here and thank Will Richardson and Sarah Covert for the preparation they put in to make this Regents Charter Lecture, Regents Professors Charter Lecture possible. And thanks to the two students, Anna Wannabe and Thomas Russell, for their introductions of our Regents Professors. And thanks to everyone for joining us today. I do hope that you will find the time to join us for such future charter lecture. So have a great afternoon and thanks everyone.